Hello, my name is Adam Velasquez,
My name is Oscar.
and we're going to be talking about bacteria growth on the skin utilizing certain moisturizers. So to begin with, we conducted an experiment with two different moisturizers using snail mucin and regular moisturizer without snail mucin. We tested alongside two control moisturizers without snail mucin, Cetaphil Daily Oil-Free Hydrating Lotion, and Neutrogena Hydro Boost Water Gel. We used agar plates in order to be used as a skin model in a controlled environment. And so over 3 days, the snail mucin containing the control moisturizers were applied to the agar plates to stimulate regular skin exposure in an open environment. The primary focus of the experiment was to observe bacteria growth and assess the impact of snail mucin in microbial environment. By comparing bacteria growth on agar plates treated with snail mucin containing moisturizers to treat with controlled moisturizers, the study aimed to analyze how the presence of snail mucin influenced microbial proliferation over the experimental period, providing a comprehensive understanding of the effects of snail mucin in bacteria growth. Now, this is our problem statement.
The investigation into the impact of snail mucin containing moisturizers on microbial growth presents a critical gap in our current understanding of skincare products. Despite the growing popularity of such ingredients in skincare formulations, there is a lack of comprehensive scientific evidence regarding their influence on bacterial proliferation. This experiment seeks to address this gap by systematically evaluating the bacterial growth on agar gel plates treated with moisturizers containing snail mucin in comparison to control moisturizers without this ingredient. The results will contribute valuable insights into the potential effects of snail mucin on the skin's microbial environment, aiding in the formulation of evidence-based recommendations for skincare practices. Now our purpose.
The purpose of this experiment is to investigate and analyze the impact of snail mucin, a key ingredient in certain moisturizers, on bacterial growth using agar plates as a stimulated skin model. By comparing the microbial environment influenced by moisturizers containing snail mucin to control moisturizers without this ingredient, our aim is to provide a comprehensive understanding of how snail mucin might contribute or to inhibit bacterial proliferation over a 3-day period. This seeks to contribute valuable insights to the field of skincare science, offering evidence-based information that could inform consumers, dermatologists, and skincare product developers about the potential effects of snail mucin on skin bacteria growth.
So, our research question was, how does the presence of snail mucin in moisturizers influence bacterial growth on agar gel skin models when compared to moisturizers without snail mucin?
And after much debate, our team came up with the hypothesis that for this research project, the skin models treated with moisturizers containing snail mucin will inhibit lower bacterial growth compared to skin models treated with the moisturizers lacking in the material.
So our procedure going forward with this experiment was to first gather materials, collect all required moisturizers, agar plate gel materials, bacterial culture, and laboratory equipment. This was led up to following standard protocols to prepare the agar gel plates using agar powder and sterile water, pouring the agar mixture into sterile petri dishes and allowing it to solidify. Next, we ensured that all swabs, pipettes, and other equipment were properly sterilized using appropriate methods. Then, we labeled the plates corresponding to the different moisturizers and the control groups. We then used pipettes to precisely apply the standardized amount of each moisturizer to the designated areas of the agar plates. We ensured consistency in application across all plates. Next, we placed the inoculated and moisturizer-treated plates in an incubator at an appropriate temperature for the growth of skin bacteria.
Now on to how we monitored and had data collection. So we did a daily observation, regularly observing the agar plates for any visible changes on bacterial growth or inhibition zones. Then we recorded our data, noting any differences in bacterial growth patterns between plates treated with snail mucin containing moisturizers and control moisturizers. Then our third step was to quantify bacterial growth using a colony counter to quantify bacterial colonies on each agar plate. Step 4. Statistical analysis. Perform statistical analysis to determine significant differences in bacterial growth between the snail mucin containing moisturizers and control moisturizers. Then finally, to conclude our experiment, we summarized our findings, noting any trends and significant observations, preparing our report to compile procedures, data, and results into a comprehensive report. So now finally, our results. we came up with a different result that didn't match any of our hypothesis or any research that we came up with, because we realized that the more moisture there is, the higher bacterial growth there will be. So we had one poor moisturizing snail mucin and then one very good moisturizer without snail mucin. And they both ended up having the same amount of bacteria, regardless of the key component of snail mucin. So we had one of each. We had one very high concentrated bacterial growth in snail mucin and one very high concentrated without snail mucin.
Our results differed from our hypothesis, and instead of snail mucin having significantly less than a regular moisturizer, we concluded that the more moisture there is, the more bacteria will grow. The moisturizers that had higher moisture in general cultured the most bacteria. For further research, we would like to try our experiment on wide varieties of moisturizers with and without snail mucin. We would also like a more accurate way of measuring our results, as well as having more time in order to do the experiment. That concludes our research video. Thank you for watching. Bye.
Thank you.